welcome back. And the last part of this afternoon session, we're going to start off with Todd Cade, who's also the chair of the session. Todd is researcher at Washington University at St. Louis, and probably has been responsible for an awful, if not 90% of the work that's done on Barth syndrome in a clinical sense. So he's an extremely valuable member of our research community, a member of our Scientific and Medical Advisory Board. And I think most of the people have wandered in, Todd, so
Good.
It's up to you now.
Great. Hopefully everyone's all caffeined up and a little more awake than I was starting to feel at the end. So what I'd like to do in the next 20 to 30 minutes is just give you an update on some of the studies that we're doing at Washington University regarding nutrient metabolism or substrate metabolism, and hopefully have time a little bit to update you on some of our exercise studies. Let's start my timer here and make sure I keep myself on time as well. Okay, so we began, back in 2006, Barry Byrne invited me to come down to one of the Orlando conferences and help them with an exercise study. So this is one of the initial exercise studies, trying to quantify exercise intolerance in Barth syndrome and see what were the mechanisms. Was it a heart-related issue? Was it a skeletal muscle-related issue, or a combination? So when I came down and started working with this group, I'd noticed the phenotype of the Barth guys, which was low skeletal muscle mass, exercise intolerance, things like that. And I thought, at the time, I was doing a lot of substrate metabolism studies at Washington University in a different population. I was actually working with people with HIV. And I thought maybe using these same techniques in Barth syndrome to really help learn a little bit more about the disease. So I initially submitted and received a Barth syndrome grant just to really look at using stable isotope tracer methodology to look at whole body fatty acid oxidation, whole body protein metabolism, and glucose metabolism. So this is also spurned from some of the clinical reports, especially from Michael Shalami's group and Richard Kelly about cardiolipid and amino acid abnormalities. Some of the exercise findings, exercise RER, which is basically a measure of substrate use during exercise, indicated that potentially substrate metabolism was abnormal in Barth syndrome. And then again, this low fat-free mass or skeletal musculature suggested abnormal protein metabolism. And in other pathologies, especially in non-Barth-related heart failure, there's an alternate fuel use in the heart and skeletal muscle hypothesis that may contribute, may cause or may contribute to the pathology. Specifically, mild heart fatty acid uptake and oxidation were lower, and glucose metabolism was higher in adults with, this is non-Barth-related idiopathic dilated cardiomyopathies. This whole thing called a cardiac cachexia hypothesis, basically stating if you have abnormalities in fatty acid utilization, which we primarily use at rest and low-level activity, you might be stealing away other types of substrates such as amino acids or glucose, and this is called the cardiac cachexia hypothesis. So when we did a study, just to really briefly summarize what we found in these preliminary studies, is we looked at the relationship between heart function, and this is the strain rate, and baseline protein breakdown. So we used these stable isotope tracer methodologies, which I'll explain a little bit more later. But we looked at there was a tendency, this is only in 5 Barth and 5 controls, a tendency for a relationship between lower strain and higher protein breakdown, whole body protein breakdown. So this was the preliminary data that we used to obtain an R01, which we're on our second year of our R01, looking at heart and skeletal muscle metabolism, energetics, and function in Barth syndrome. And really, our overall hypothesis is what both heart and skeletal muscle fatty acid metabolism is impaired in Barth syndrome, which facilitates increased heart and skeletal muscle protein catabolism, potentially supplementing with amino acids for energy, and enhanced glucose metabolism, again, to supplement for this reduced fatty acid metabolism in order to supply the energy needs. And because amino acids and glucose provide an inherently lower amount of ATP than fatty acids do, an energy deficit occurs, and this may contribute to impaired energetics of the heart and skeletal muscle, which may mediate or at least potentially exacerbate the exercise intolerance that we see in this group, fatigue, muscle wasting, and heart failure in Barth. And that was really our overall hypothesis. So what we did we wanted to basically characterize, do a cross-sectional study, and characterize cardioskeletal nutrient metabolism. So we looked at children and adults. So we looked at 15 children. Well, we are looking at 15 children. We're proposing to. I'll show you some data, not quite that many yet, with Barth, and compare them with healthy age, BMI, Tanner stage, and activity level as much as we could, match controls in adults, a total of 60 participants over 5 years. So our first aim was to characterize skeletal muscle fatty acid oxidation and lipolytic rate, so fat breakdown rate, amino acid, proteolytic and oxidation rate. So we use actually a leucine tracer as kind of a measure, our general mark of protein metabolism, and glucose. So we looked at glucose disposal and hepatic production rate. During rest, so we did a 2-hour resting program, so basal kinetics. We also did a low-intensity exercise bout of 30 minutes, and basically what we did is we did peak exercise testing. We measured their peak exercise level, and we did so over peak VO2, and then we exercised them at 50% of their peak VO2 for 30 minutes. And then we did an hour post-recovery, and we looked at kinetics, whole body kinetics of amino acid and glucose and fatty acids in both participants in Barth and controls. Our second aim was to actually look at not whole body metabolism, but look at specifically heart fatty acid, amino acid, and glucose metabolism in adults. So we used PET imaging, which I'll explain, radioactive isotopes and PET imaging. So we didn't look at this in the children. So we just started doing that. And the methods that we used were stable isotopes, so stable isotope tracers. So what we did is we basically infused stable isotopes of glucose, leucine, and palmitate, and then by taking blood and breast samples and putting them on a mass spectrometer, we can tell a lot about their kinetic rates. Fatty acid oxidation rate, amino acid oxidation rate, how well they're utilizing glucose, how much protein they're breaking down, things like that. And then with radioisotopes and PET imaging, we can actually look at specifically how the heart is able to utilize palmitate, leucine, and glucose. And then our second specific game was to actually look at these kinetics, so stable isotope kinetics and the radioisotope kinetics, and look at the relationship to energetics and function of the heart and skeletal muscle. So what we did is also we did magnetic resonance spectroscopy, so 31P phosphorus, looking at the energetics of the heart and skeletal muscles in the magnet. So we looked at energetics that way, we looked at function of the heart through echocardiography, and then we looked at skeletal muscle function through exercise testing, looking at peak VO2. Okay, and then lastly, what we did, I'm not going to talk too much about this, but what we're doing is so we're looking at whole body and tissue substrate metabolism and function in these guys. And then we also wanted to look at molecular pathways of nutrient metabolism. Specifically, again, what we're looking at, protein catabolism, oxidative phosphorylation, fatty acid metabolism. And our first proposal was to actually get skin biopsies. But after the first few subjects and who had really low ANCs and a couple kids refused, we decided to go another route. Actually, it was from Barry. It was a suggestion from Barry, who's collaborating with us with this, to use renal epithelial cells. There's a paper that came out to be able to have, from urine, to collect these renal epithelial cells and induce them into iPS cells and then differentiate them into cardiomyocytes or whatever. So we took that suggestion, and our core group in genetics was able to really do that. And so we were able to induce these urinary epithelial cells into iPS cells. And we just recently, a couple weeks ago, sent them to Barry's group at Florida. So this is a picture. This is actually a recent picture, just a week ago. We did it. This is a picture of one of our clinical studies. This is our GCRC or CRU nurse that helps out here. So this is one of our participants. You can see up here, this is a fatty acid, so palmitate and albumin and glucose and saline, and basically infuse these stable isotopes by taking blood samples. As you see here, this is a warming box that helps us get arterialized blood, or helps us draw blood a little easier. And he's actually going through the exercise session right here on our recumbent bike. We also get breast samples, and here's the RMR cart behind us as well that can help us quantify VCO2. Okay, so what I'm going to just show you the data thus far is we didn't show any. I'll show you a little bit of the adults, but we just started testing the adults in January because one of our tracers was just validated in January, and we just started testing adults in January. So far we have data from 10 control kids and 7 Barth, average age about 12. As you see, the weights are less, not super surprisingly. Fat-free mass is significantly lower in the Barth group, which we've shown before, which is, again, not surprisingly. Fat mass, there's no difference, but when you look at fat mass as a percentage of body weight, it's significantly lower in Barth. And as no surprise, absolute neutrophil count was lower in the Barth group. Peak VO2 significantly lower, about a little less than half of what their peers were. Ejection fraction really in the normal range, just a little bit lower, and fractional shortening on the low normal range. So this first slide shows you palmitate oxidation rate. So again, this is a 13C labeled tracer, so the label's on the carbon. So when you oxidize palmitate and it goes through the TCA cycle, you spin off the carbon labeled on the palmitate, actually comes off and it comes out as CO2, and you can collect it in the breath and measure it. So knowing the enrichment of palmitate in the blood, knowing the enrichment of the label in the breath and 13CO2, you can tell palmitate oxidation rate. So again, we measured this at baseline during this low-level 50% peak VO2 exercise and then recovery. So as you see, there's no differences during baseline, and again, this is what we showed actually earlier in our first study. No differences in palmitate oxidation rate during baseline or recovery, and it seems like there's a tendency for a lower palmitate oxidation rate during a low-level exercise. Again, so if you think about if you're not with exercise physiology, 50% of peak VO2 is considered probably a low, moderate exercise. So for us, it would be probably a brisk walk. So when we looked at leucine rate of appearance, and this is basically a surrogate marker for whole body protein breakdown. Again, same conditions, baseline exercise and recovery versus control and Barth. You see there's a tendency for higher whole body protein breakdown at baseline during exercise and recovery. So during all these conditions, it seems like the boys with Barth seem to be breaking down more protein. But what's interesting is when you look at leucine oxidation. So where is this leucine going? Is it going to make energy? There was really no differences. So this protein breakdown, this leucine breaking down into the body, it doesn't seem to be oxidized at baseline. Really doesn't seem to be much here as far as rest or recovery. So again, this is preliminary data, but right now it doesn't seem like this increased protein breakdown is being used to make energy. So I think this is one of the more interesting findings so we looked at glucose disposal rate. So basically this is just how much glucose is disappearing going into the muscle and being utilized for energy, which we think it could be stored as well, but we think it's energy. So again, same conditions, and if you see the boys with Barth significantly at baseline or basal levels, exercise and recovery are using significantly more glucose during all these conditions. So it seems that they are compensating for potentially this reduced fatty acid oxidation by compensating with increased glucose. Oh, getting a little bit. Okay. And so what we did is we also measured plasma lactate concentration during the same conditions. Plasma lactate concentration at baseline and recovery, no different. But as you see, at a low level of exercise, plasma lactate is at a fairly high level. If you're familiar with exercise physiology, when you and I are at peak exercise, we're probably right around 7 up here when we're absolutely exhausted. We cannot go any further. So at a very low level of exercise, the guys with Barth have a fairly significant lactate level. So we also, when we looked at skeletal muscle energetics, so what we looked at in the magnet was this PCR recovery. So it's an indicator of muscle oxidative phosphorylation. So we're looking at the recovery after you do a little exercise bout. So we have a little machine that's MRI compatible. It looks like a gas pedal. And what the boys do is basically push on this gas pedal to fatigue their calf muscle. And what we do is quantitate the PCR, the concentration of phosphocreatine, And then once it basically gets depleted with exercise, and then when they stop exercising, it recovers, and it's typically an exponential curve. And we can calculate basically ATP generated from oxidative phosphorylation. So as you see, the black is the Barth subjects, and the white are the control. As you see, just the general levels, okay, of oxidative phosphorylation are lower in the Barth syndrome group. And it seems to be significantly associated with exercise tolerance. This is peak VO2. So the direct correlation between skeletal muscle oxidative phosphorylation and whole body exercise tolerance. Okay, so then we also looked at a different marker, but using PCR ATP ratio, which is a good predictor of energy reserve or oxidative function in heart in these boys. And again, if you look here, the black is Barth syndrome, the open circles are the controls. So the levels are significantly lower, PCR ATP ratio lower in Barth syndrome than in controls. And it seems to be related to fractional shortening. Okay, so indicating basically the oxidative reserve in the heart seems to be directly related to the function of the heart. Okay, so we just got some images from the PET imaging, and this is in one adult. So this is just a representative slide. So we've tested 3 adults with Barth and 5 controls so far, but we don't have actual data yet. But I wanted to show you a little bit just within one control and one patient with Barth. So this is basically using a C11 palmitate, so it's a radioactive tracer, and using PET imaging. And as you see, this is basically the myocardium. And when the heart takes up the palmitate, it basically gives off the positrons, and the PET imager is able to detect that and give us real pretty pictures. And through by scintillation counting C11 CO2 and different metabolites, we can tell a lot about the metabolism. So hopefully in 2 years I'll have a lot more data. But this might be difficult to see. So the blood line is actually the blood radioactivity, or the blood activity count of the C11 palmitate. And so again, this is control, this is Barth. And it's probably hard to see. The blue is tissue, so I want to kind of focus on the blue. And it's probably difficult to see, but what you see here in the control, the heart is able to take up the blue, the tissue is able to take up the palmitate, and it clears it relatively quickly. But as you see here in the Barth participant here, the blue line does not go up like that this way. So it doesn't seem like the heart is taking up as much palmitate. And it seems to hang around in the tissue a lot longer, so it's not cleared as fast. Less is coming up and less is being cleared, it seems like, in the heart of the Barth participant. Again, this is one patient. We don't have actual data yet, but I just wanted to kind of give a flavor of what we're doing. So this is C11 leucine. Again, a representative picture here. It seems to be the same trend. Again, this is a control. This is a Barth syndrome patient. And as you see, again, the blue line uptake in the control patient in the heart pretty quickly, and then it clears it relatively quickly. But it seems to be blunted here in the Barth syndrome participant. And again, it kind of hangs around again in the myocardium a little longer than it does in the control patient. And then lastly, there's not a great deal of glucose uptake, it seems like, and it doesn't seem much different in the Barth versus the control. But this is pretty standard. The heart pretty much uses fatty acids during rest, and unless you give it insulin stimulation, unless you have a meal, there's not a whole lot of glucose uptake in the heart, typically anyway. Okay. And so I wanted to present, I just actually got these data sent to me yesterday. And this is a separate study. This was a smaller study of 3 participants. This was actually a study that I buddied on, an exercise training study, which we looked at. We wanted to look at arginine and citrulline kinetics, obviously from all the work from Dr. Kelly and supplementation. We want to say, why is arginine potentially low in these subjects with Barth syndrome? So what we did is we did the same thing. We basically used a couple different tracers. We used arginine and citrulline tracers, and we wanted to see the kinetics in basically breakdown of arginine and clearance of arginine, and see if there was any differences between the Barth syndrome participants and not. So again, these are young adults, N of 6. So there's three controls and 3 Barth patients. And again, this is very preliminary data. So when we looked at arginine rate of appearance, so this is basically coming into the plasma, so representing breakdown of protein and release of arginine into the plasma, it's significantly, again, this is only 3 subjects in each group, significantly higher in Barth syndrome. So we do not have plasma levels yet. We just ordered a standard for that. But we assume that the plasma levels of arginine are going to be lower too, which accompanied with these data would show that not only the rate of appearance or breakdown is higher, but clearance will probably be higher also in the Barth participants. And then citrulline rate of appearance, again, so there was no difference in citrulline rate of appearance in this group. So basically, we don't know exactly the fate of arginine, so what we know it's breaking down and getting cleared probably faster, but where it's going, we don't know. We have to do more detailed tracer studies. But I wanted to just show it to you because these are the first data that I know of that show actually differences in arginine metabolism besides just plasma levels in Barth syndrome. Let's see where I am here time-wise. Okay. What's that?
It's about time for questions.
Okay, well, I'm only at 20 minutes, but okay. I'll go with the boss. Okay, so preliminary conclusion here. There's a tendency for lower fatty acid oxidation during exercise in Barth syndrome. It seems that this is supplemented with higher glucose utilization during rest, exercise, and recovery. Leucine metabolism, there's a lot of variation, so that's a little bit less clear at this point. Lactate production is higher during exercise and even low-level exercise. Again, they are able to tolerate a pretty high lactate production during low-level exercise. Heart and skeletal muscle oxidative function and energetics measured by MRI is lower in Barth syndrome and appears to be related to heart function and exercise capacity. Looks like there's some differences, again, this is just in one participant versus one control, but differences in myocardial fatty acid and leucine metabolism between the two. And then higher arginine RA in Barth syndrome, clearance is likely higher as well, but we need to obviously do some more studies with that. Okay, so I am just going to go skip the exercise stuff, and we'll take some questions. Just want to thank, okay. So I certainly want to thank Cade, who is my research coordinator here, who basically organizes these studies and kind of makes them flow nicely. And there's a lot of people. I'll also recognize Barry Byrne, who's been really an incredible help, and he's assisting with this study as well as with the IPS cells. And Carolyn Spencer, who initially was involved in these studies as well. And there's a whole slew of people at WashU that helps, and then obviously the funding from the NIH. And then a special thanks to the Barth Syndrome Foundation, who really seeded many of these studies, and then the participants that come from all over the U.S. and all over the world, and especially the U.K., to participate in these studies. So thanks for your attention.
Whoa, thank you, Todd. That was wonderful. Am I okay now? Okay, maybe I need another mic here. Looking for questions here. If not, I'll ask a question. So, yes, here you go.
It's a very nice talk. You talk about glucose utilization is greater during exercise. Can you comment about whether or not it's being fully oxidized in the TCA cycle? You've got a high lactic acid production, so it's not entering the TCA cycle. You're just using it through the glycolytic pathway, and then it's just being shunted off as lactate.
It's a great question. So we could measure glucose oxidation for sure, but when you use a tracer, you can only use one 13C labeled carbon tracer at one time, because when you collect it at the breath, you can't tell if you give two at one time, they both come out at the same time. Right. So we are doing two separate studies looking at palmitate oxidation and then leucine oxidation, so that would be a separate day. But certainly, I would probably hypothesize that it's not so much getting oxidized, so it's either...
I think that's right.
Yeah, I think it's probably either lactate going gluconeogenesis or some kind of the Cori cycle, or however it's probably being used glycolytically, and then the lactate's probably being shuttled, and potentially making new glucose. That would be my guess.
I think that's exactly the way I think too. That the cardiolipin defect is causing a problem with the electron transport chain, and then that fouls up the ability of the TCA cycle to run because of the buildup of NADH. can't oxidize fatty acids, and the glucose can only go part way.
Right.
And then you show a decrease in arginine. And, well, actually, you didn't show a decrease. You showed an increase, but you're suggesting there's a decrease in the plasma levels, and that was shown by the previous author and by Dr. Kelly.
Right.
What about the idea that the arginine is being, I think that arginine can go into creatinine or creatine, is a precursor of creatine phosphate, and also could be making nitric oxide, which maybe is a compensation by these individuals in trying to increase blood flow to the heart or to other tissues.
So Dr. Kelly can probably answer that first question, but the nitric oxide question, basically having these two tracers, we're able to calculate nitric oxide production, and hopefully next time I present, we'll have those data and look at. So basically, we modeled this infusion protocol off of a protocol coming from Baylor, looking at nitric oxide production in critically ill septic patients. So we're actually able to look at these data too. So we're going to be able to look at nitric oxide production in Barth syndrome as well. So I don't think anyone's looked at that yet. So I don't even know what to say, which way it would be. Yeah.
Other questions, please? Richard, comment?
Check.
Just to answer that question, about 20% of daily arginine consumption goes into phosphocreatine or to creatine synthesis. We find it takes 2 to 3 times the daily intake of arginine to get the level close to normal. So that's some idea of the flux.
I don't know if I could do that with two mics here. Does anybody have another question? Because I have one. Okay, it seems to me that the boys are glycolytic, and you've shown that. Their heart can pump enough blood to their muscles, and so, and yet they can't seem to extract oxygen from that blood and turn it into, go all the way down the TCA cycle. So, does that make sense to you? They must metabolize some sort of fatty acid at some point. They're not devoid of fatty acid oxidation to some extent.
They're at rest it's normal,
Yeah.
but at exercise they're still metabolized. They're oxidizing fatty acids, just not as much.
And so the defect occurs at the step of exercise. They just don't click on to that fatty acid oxidation to the level that a normal individual would.
That's what it seems like,
Yeah.
right? So where the backup is, again, we talked about, it could be the different levels of ADP and it can inhibit fatty acid oxidation and it could be compensating. There's a number of different, we haven't looked at those specifically, but that's at the level of.
Okay. Yeah.
Just to add to this specific point, I mean, the data can be explained simply by having less mitochondria, right? Because having less mitochondria, you're diminishing your maximal beta oxidation capacity. I mean, I'm not sure if this is true, but this would be one simple explanation.
Well, having less mitochondria would have a lower peak VO2 and probably less fatty acid oxidation, but I don't know if that would be compensated then by, well, I guess it potentially could by glycolysis, but that's potentially true as well.
Have you ever measured a pH in blood or muscle? Because lactate is increased, so that may change the pH too.
Sure. No, we haven't. We haven't looked at muscle pH or arterial gases or anything like that. I think this is the first time lactate was measured after exercise anyway in Barth syndrome, to my knowledge, at least in humans, to my knowledge.
Todd, I can comment because we did that once with bedside exercise in a patient with a central venous catheter, and the pH was markedly reduced after mild exercise because of the lactate. There was just one, N of 1.
There's a question in the corner.
Thanks, Matt. I know, hiding in the dark. Have you ever looked at plasma creatine kinase pre and post exercise?
No, we haven't. So this is the first by MR we're looking at it in the heart and then looking at it in skeletal muscle. We have not looked at it in plasma, but potentially Richard has, or someone else has. We have not. Actually, I take that back. We looked at CK pre and post our resistance training exercise, and I can't recall the values, but what I can tell you is that the exercise, at least chronically, did not increase plasma CK.
Yes, we generally don't see any increase. I haven't done exercise testing, but it's usually normal.
It was a normal level, right?
If,
Yeah, it was normal.
if not a bit low.
Okay. Any other questions for Todd?